Okay, hello again. We are going to see now the plasma membrane. Here you have what we will review today. Structure and function of the cell membranes, the different types of cell junctions, and in a different video we will see the transport again through the membranes, which is well, a bit complex. The plasma membrane, which has many important functions besides of being a boundary, is so important that any disorder of that membrane may cause the cell to die. But first, let's see an animation of the plasma membrane.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
Okay, besides the plasma membrane, the cell has many other membranes in its interior. The general function of membranes are very important. So let's review those functions. The first one you have there in the screen, the cell membrane is a semi-permeable barrier. So that means that it allows some molecules to go through it, but it retains some others. It also protects the inner environment. Inner membranes help to create different compartments. Membranes can also regulate transport through them. The cell membrane can function as a receptor to different signals like hormones. They also permit cell recognition. They provide anchoring sites to the cell skeleton and the external lining known as the extracellular matrix. They also have special proteins known as enzymes that facilitate chemical reactions. They also facilitate the passage of molecules from one cell to the neighboring cell. And they also participate in membrane fusion and motility. So as you see, there are many functions, many important functions of membranes. As seen before, phospholipids are arranged in a bilayer where the hydrophobic tails are located in the center away from water, and the hydrophilic heads are located in contact with the inner and outer water. Given that it is a quasi-fluid structure, its components are in constant free translatory movement. In animal cells, most membranes are made of 50% lipids and 50% proteins. We can say that the primary components of membranes are phospholipids, which are amphipathic because they have a hydrophilic side and a hydrophobic side. the hydrophilic sites are the ones that are in contact with water. The fact of being amphipathic forces the phospholipids to arrange in that characteristic bilayer. This model is called the fluid mosaic and was first proposed by Singer and Nicholson back in 1972. And also you have to know that, of course, the plasma membrane is a very, very thin structure. So as you see in the box, yellow box below, it's only 7 nanometers. Phospholipids are not the same type in the inner and the outer half of the plasma membrane. The inside half is made of phosphatidylserine, phosphatidylethanolamine, and phosphatidylinositol, while the outside half is made of phosphatidylcholine and sphingomyelin. Located among the phospholipids, we have cholesterol, who has an important influence in the fluidity of the membrane and can make up to 50% of some membranes. Finally, and only in the outer half of the membrane, we may find up to 2% of glycolipids. This image, these 3 images are to show you the chemical structure of phospholipids, although you don't have to know that. It's just informative. Going back to cholesterol, you need to know that it is located among the hydrophobic tails of the phospholipids. And besides contributing to the stability of the membrane, its main function is to regulate fluidity of the membrane by preventing the tails to pack together. That may happen when the temperature drops. If you ever seen oil in cold, it gets like thicker. But in this case, it also prevents excessive fluidity when temperature rises, for instance, when you have fever or is too hot. Most of the functions of the membrane are carried out by proteins, so it is important to know the different proteins found in membranes. Alright, so let's go with it. As said before, proteins made up to 50% of the weight of the plasma membrane. We can divide those proteins into 2 big groups depending on their location. Those that cross or penetrate the membrane are called integral proteins, while those that don't get in contact with the hydrophobic core are peripheral proteins. Now, let's see some of those proteins. Okay, ion channels are made of integral proteins that cross the membrane side to side, as you see in this animation. And they form water-filled pores for the passage of specific ions, which are very, very tiny particles. Membrane carriers are integral proteins that may change their shape to transport specific molecules like amino acids. You know that proteins are made of amino acids. And these are also called transporters, this type of carriers. Another type of integral proteins are receptors. These can bind specific molecules that then trigger a process that carries out a function of the cell. The most common example are hormones or neurotransmitter receptors of the neurons, the cells of the brain and the nerves. Okay, then enzymes are specific proteins that may accelerate or catalyze chemical reactions. These can be found associated to membranes as integral but also as peripheral proteins. Then we have linker proteins that can also be integral or peripheral, and their function is to anchor filaments that make up the skeleton of the cell, but they also anchor filaments in the outside of the cell. So 2 cells together or or they also may participate in the movement of the cell. Some proteins may have sugars linked to its outer side and are then called glycoproteins. Cell identity markers belong to that group of proteins, glycoproteins, and their function is to distinguish self cells from foreign cells. So in the case of a foreign cell, it's going to be attacked by the immune system. An important marker present in most cells in your body is the major histocompatibility complex that acts as a passport so the cell is not destroyed. We may also find glycolipids that together with glycoproteins are involved in recognizing cells. And then I wanted to talk about another example of glycoproteins. Those are CD antigens that are found in the membranes of lymphocytes, which are the main cells of the immune system. So how that thing works? Well, here you have the structure of the major histocompatibility complex used by cells to distinguish self from non-self. Then of these you have MHC1 of your own cells that can link to CD8 of your lymphocytes and be identified as own cells. Then we have another type which is called MHC2 that can link to the CD4 protein of your lymphocytes and also be identified as a self cell. So in both cases, any cell with your MHC1 or 2 will not be attacked by the lymphocytes. But a foreign cell will have different MHCs, and your lymphocytes will not identify it as self cells, and they will destroy them. Okay. Similar cells make up tissues, and for that to happen, they need to interact. For that to happen, cells secrete a complex of proteins, fibers, and carbohydrates around them that we call that the extracellular matrix. This extracellular matrix is made of collagen fibers that provide resistance, proteoglycans that make that matrix viscosity, so it feels like a gel, and also has fibronectins which stick together the fibers of collagen and proteins of the membrane, in this case integrins, so the matrix remains strongly attached to the cell's surface. All right, in this image you can see a layer of cells, and you see cells are organized to form tissues, and tissues form organs, and all the organs together form an organism, in our case a human body. For this to happen, cells must first stick to each other firmly, and this is possible thanks to specific types of integral proteins known as cell adhesion molecules, or CAM. Among CAMs, cadherins are responsible for adhesion among similar cells. We call that homotypic interactions. Next, we will review the different types of cell junctions, and you will see that cadherins are found in desmosomes and adherens junctions. On the other hand, we have selectins, which are responsible for heterotypic interactions, which involve different cells, different types. Okay, and now let's see the different types of cell junctions. There are not too many. What are cell junctions? Well, they are points at which two plasma membranes attach to each other to maintain the cohesion of a given tissue. Here the picture in the right shows three enterocytes, which are the absorption cells in the inner surface of your small intestine. The white area is the inside of the tube where the food is located during digestion. For the food not to move in between the cells, we need a tight junction that fuses together the two membranes. If you see in the two other images, you have the microscopic aspect of this type of junction. It looks like a zipper. Adherence junctions have a plaque of dense proteins in the inner aspect of both plasma membranes. You can see them as blue dots in the first image. And these attach to the membrane and to the microfilaments of the cell's skeleton. Cadherins are transmembrane glycoproteins that insert into the plaque inside and also connect to the opposite cadherins of the neighboring cell. This type of junction is abundant and forms adhesion belts that resist separation of superficial epitheliums. Then desmosomes are similar to adherence junctions because they also have a plaque with cadherins, but in this case they bind to intermediate filaments of the cytoskeleton, which are made of keratin. The intermediate filaments extend through the cell and bind to desmosomes on the opposite side of the cell. Desmosomes also prevent separation of the cells and can be found in the epidermis or in the heart. And we will have a lecture on the cytoskeleton so you get to understand all this better. Hemidesmosome, the name means half desmosome, so they look like half desmosomes, but their glycoproteins are integrins instead of cadherins. Their function is to anchor cells to the basement membrane, which is a membrane outside the cells. Okay. As desmosomes, they attach also to intermediate filaments of keratin inside the cell, but outside they attach to the laminin of the basement membrane. Then we have gap junctions, which are made of connexins that form tunnels called connections or connexons, through which cells can share ions and small molecules, but also collaborate to the spread of chemical and electrical inputs. So you can find them in the nervous system, the heart, or the uterus. Okay, during labor, the uterus has to contract. This image is to show all those type of junctions. Okay, as you see, a same type of cell can have more than one type of junction. In the right you have again the two enterocytes showing three types of junction at different levels. And this is all for junctions, so don't worry. By now, if you don't fully understand something, it will become clearer as you progress and know more about the cell.